Maybe suggest
Okay.
people settle down.
All right. Okay. Okay, everybody, we should get started. Welcome to session 2, and David Sullivan will introduce our keynote speaker.
Thanks, Fotini. It's my privilege and pleasure to introduce our respected colleague and mentor, Dr. Dan Goldberg, the David and Paula Kipnis Distinguished Professor of Medicine and Molecular Biology. He's been a superb scientist that has made keen observations, which he has tracked down to details. One of the big lessons he taught me was you've got to support it in many different ways. He first started looking at ordered hemoglobin degradation, followed it up with brute force purification of the aspartic protease, now called plasmepsin. And he's valued science as much as he's valued mentoring, and he's been superb at both. Dan, thanks. One last comment. Although I was sort of trained to consider the digestive vacuole the center of the plasmodium universe, I think he's going to talk outside the digestive vacuole today to talk about the galaxy. Thanks.
Great. Thanks very much, David, and thanks to the organizers for inviting me here. It's a great symposium to be able to participate in. Okay, so you've already seen this slide from, I believe, Manoj and Regina, and this is the beautiful red blood cell. It's a lovely biconcave disc, optimally suited for oxygen transport and delivery in the bloodstream. And then consider what happens when it gets infected by a malaria parasite. Now it's lumpy, bumpy, misshapen, and a lot has happened to transmogrify this cell and for the malaria parasite's nefarious purposes. So this all starts, as you've heard from Manoj, when a merozoite attaches to the red blood cell and pushes its way in, and it seals itself with a piece of the host cell membrane to form a parasitophorous vacuolar membrane, which is mostly from the red cell, but the parasite contributes to it. It secretes rhabdoids and dense granules to add its own proteins and lipids to the process. And then it exists inside this vacuole, which is inside the red blood cell. So what are the pros and cons of living in a vacuole? Well, it's a good thing for the parasite because it's a nice, warm, cozy environment full of what it needs to grow. It's buffered and it's relatively sheltered from the immune system. But it has some challenges because it has to, let's see, did we? Yeah, thank you, okay. So it has to get proteins out into the host cell through all these membranes in order to change the red blood cell for its purposes. And it has to get nutrients in. And so those are the two challenges that it has. And in terms of getting nutrients in, what it has figured out to do is to export proteins into the red blood cell to set up a nutrient channel called PSAC, Plasmodium Surface Anion Channel, that takes things in from the outside. And this has been well characterized by Sanjay Desai's lab. And then it also has to get things from the red blood cell into the parasitophorous vacuole, different nutrients, so that it can then take them in through specific transporters, such as the glucose transporter and the nucleoside transporters, and for its growth and maturation. And if you look under the hood at what's going on in the infected red blood cell, the parasite is putting out all sorts of things. It sends out several hundred proteins, exports them into the red blood cell. They set up these knobs that then display the PFEMs that Kirk told you about earlier so elegantly, and that is involved in cytoadherence. It makes Morris clefts that are Golgi-like way stations that sort proteins to different destinations in the host cell. It makes vesicles like K dots full of kinases, J dots full of chaperones. There are soluble proteins floating around. It alters the cytoskeleton of the red blood cell, and it's really doing all sorts of things. It even sends out exosomes ostensibly to communicate with the host cells as well as with other parasite-infected red blood cells. So out in the red blood cell, it is taking in nutrients and also exporting wastes. It is mediating cytoadherence to avoid the spleen, as we heard. And it is modifying the cytoskeletons for better rheologic properties in the bloodstream. How does it do all this? Well, so it makes these several hundred exported proteins in the endoplasmic reticulum, in the parasite's own ER, and then sends them through the secretory system out into the parasitophorous vacuole. And there, they have to be translocated by an apparatus called PTEX, Plasmodium translocon of exported proteins, that was first proposed by the Crab and de Koenig-Ward group. And this complex recognizes somehow, mysteriously, I must say, proteins destined for export and sends about into the red blood cell. We now have a structure of the PTEX complex from the beautiful work of Mimi, who will tell you more about this. I'll certainly leave it for her. But there are three core components: HSP 101, a chaperone, PTEX 150, a linker protein, and EXP2, the channel protein. We wanted to study the functionality of these components, and so what we did is we made a regulated knockdown of the HSP 101 chaperone protein, and when we did, we found that under permissive conditions, our favorite exported protein, histidine-rich protein 2, got fully exported into the red blood cell. You can see the red blood cell here and the little parasite inside. But under knockdown conditions, when HSP 101 was depleted, the exported protein accumulated in a rim around the parasite in the parasitophorous vacuole. And the same was true of a Morris cleft protein. This is its typical distribution, but when HSP 101 was knocked down, it was in a ring around the parasite in the parasitophorous vacuole. Similar results were obtained by the Crab and de Koenig-Ward group. with the linker protein, PTEX 150. And we examined the third component, EXP2, the pore protein. And what we found was that, again, when under knockdown conditions, the export of HRP2 or a Moore's cleft protein, SBP1, was blocked. But these parasites had a slightly different phenotype. The timing was different from the knockdowns with the other two PTEX components. We actually, the phenotype of all three leads to death, but the stalling for the other two components is at a specific ring trophozoite transition, whereas the EXP2 knockdowns could lead to death at different points in the intraerythrocytic life cycle. And so that was a clue to us that maybe it had a different function, as was the fact that we found a stoichiometric imbalance between the synthesis of EXP2 and that of the other components. And so we remembered back to a paper that was published by Sanjay Desai when he was a graduate student at Washington University in St. Louis, where he physiologically characterized a channel protein, this green protein here, right here, and found that it was a non-selective pore for small molecules, small nutrients across the parasitiferous vacuolar membrane. And so we wondered whether EXP2 could be the molecular identity of this pore. And so we collaborated with Matthias Garten in Josh Zimmerberg's group at the NIH. And Matthias was able to extrude parasites with the parasitiferous vacuole intact from the red blood cell down here. And he was able to patch clamp, which is really a heroic effort, and obtain recordings that showed a channel activity similar to what Sanjay Desai had seen 25 years earlier. And what was interesting was that he didn't see it in all patches. It was patchy, it was sporadic. But what we found when we made different parasite lines, either overexpressing HRP2 or knocking down EXP2, either overexpressing EXP2 or knocking it down, we found that we got different frequencies of positive patches with the signal of this channel. And in fact, the level of expression of EXP2 correlated linearly with the frequency of channel detection. So we suspected from this that EXP2 is the nutrient pore, that green channel that I showed you. But one more important experiment, what we did is we deleted the charged residues at the C-terminus of the EXP2 protein. That had no growth phenotype, so we were able to grow up the parasites and do the same patch clamping and electrophysiology. By we, I mean Matthias. And what he found was that there was a different gating of the channel when the C-terminal charge region was deleted. And that suggested that this really is responsible for the channel activity per se. So EXP2 appears to function as both a protein export translocon channel and as a nutrient channel. And this dual function rang a bell because we'd been thinking about dual functions in a slightly different context, because there's a protein called plasmepsin-5, an ER aspartic protease that actually processes exported proteins in the ER co-translationally at a conserved pexyl protein export element motif to generate a mature N-terminus that's needed for permitting these proteins to be exported. And plasmepsin 5 also seems to have a dual function because it's involved in both protein export and in dense granule protein maturation. Remember, the dense granules are those little vesicles that are secreted at the time of establishing the new parasitophorus vacuole just after invasion. And so the dense granule protein function appears to be, at least we think it's the primordial function. It's also important in toxoplasma and a similar function. For EXP2, we think the nutrient channel is its primordial function because toxoplasma also has a similar nutrient channel using an ortholog. And so our hypothesis is that HSP101 was a pre-existing ubiquitous chaperone. EXP2 was a pre-existing channel for nutrient uptake. Plasmepsin 5 was there for dense granule proteins, and the malaria parasite evolutionarily cobbled together an export system from these components using an adapter protein, PTEX 150, to link them all together to be able to export proteins and further its pathogenesis. So I've talked to you about the functions that these exported proteins have in out in the red blood cell, but the parasite has influences beyond the red blood cell, particularly in endothelial dysfunction. And as you saw, mechanical obstruction is an important feature of the cytoadherence. There's endothelial activation that goes along with this, as well as with other mediators. There's impaired vasodilation. There's a block of nitric oxide synthesis. There's even a destruction of the backup molecules, the epoxyeicosotrienoic acids that mediate vasodilation when NO is low. And there's disrupted endothelial barrier integrity that creates vascular leakage, which is the hallmark of severe and cerebral malaria. So in this context, we were interested in our favorite exported protein, histidine-rich protein 2. This is its sequence. It's a remarkable protein. It's mostly histidine and alanine. And it's produced in the parasite. It is exported into the red cell when the parasite bursts out. It spills into the bloodstream and accumulates to high levels, up to 100 micrograms per mil in the bloodstream of patients with malaria. It's the basis of the rapid diagnostic test for falciparum malaria. It's only present in falciparum and related monkey malarias. And its levels in the bloodstream correlate with severity of malaria and progression to cerebral malaria. And shown here is just one study showing the dramatic elevation of HRP2 levels in cerebral malaria. So we wondered if maybe this molecule wasn't being put into the bloodstream by parasites for detection and prognosis, but rather had another function, perhaps on the endothelium. And there was a seminal study done right here at Hopkins by the teams of David Sullivan and Monique Stins about 15 years ago, in which they took infected red blood cells and showed that when they put them on an endothelial monolayer, they could get a decrease in resistance across the endothelial barrier. that was concentration, red cell number dependent. And they went on to show that it did not require contact with the endothelial barrier and postulated that there was a soluble factor. So we repeated this experiment using two different parasite strains, 3D7 and DD2. And a major difference between these two strains is that 3D7, shown in red, is it has the HRP2 gene, whereas DD2 in orange does not. And this was first shown by Tom Wellham's group. He also was one of the discoverers of the HRP2 gene in the first place. And so there clearly was a difference here. And we went on, and what we did is we put the HRP2 gene back into DD2. And when we did that, shown in blue and in green here, we could recapitulate the vascular permeability leak in an endothelial barrier. This is using human cerebral microvascular endothelial cells. And we could also recapitulate this using the parasites by adding recombinant HRP2 to the system. And we could block this with a monoclonal antibody to HRP2. So it looked like HRP2 was responsible for this phenomenon. But we had extremely variable results using recombinant HRP2. And it turned out that what we were looking at, the HRP2 made in E. coli is bound to a lot and a variable amount of polyphosphate. And polyphosphate is an endothelial barrier disruptor. And when we stripped the polyphosphate on, it was inactive. But native HRP2 purified from parasites was quite active. And when we looked to see what was bound to native HRP2, we found that there were 50 hemes per HRP2 polypeptide, which is just remarkable. That was first found by David Sullivan in a titration experiment. But what comes out of the parasite has 50 hemes in it, in that small polypeptide, which has about 98 histidines. Anyway, it's loaded with heme, and it's a large multimer, as shown by blue native page gel. And we could also look at it by size exclusion chromatography and by analytical ultracentrifugation. And that gave us an estimate of that it is a 14-mer. And you can actually see particles of it by negative stain EM. And you can see it in the blood of patients with falciparum malaria, shown here and here. And here's a blue native page gel again, using a Western blot for HRP2, or using a fluorescently conjugated hemopexin, which is a heme binding molecule, very tight heme binding molecule, which also lights up the HRP2. So we think that this is, we estimate it's a 14-mer. Each polypeptide has 50 hemes, so that means these nanoparticles have about 700 hemes each. What about, they bind heme, about 50 hemes per polypeptide, either native or recombinant. You can strip the heme off, and then you don't detect any heme. And you can make a mutation in which you delete the last 4 C-terminal amino acids, and then it will still bind heme fully, but as I'll show you, it no longer binds to endothelial cells. So, and it's a combination of a monomer and a trimer when you have this mutation. So you appear to need the full multimeric particle for binding to endothelial cells. And you also need the full particle for activity. So when we measure the endothelial response, here we're not looking at barrier disruption. We've got an easier assay. We know that HRP2 triggers interleukin-1 beta, so we made an interleukin-1 beta venous fluorescent protein reporter line in the human brain microvascular endothelial cells. And you get stimulation of IL-1 beta release with the heme-loaded molecule, but not with the unloaded or the non-binding versions. And the ligand is very important for this. So here's HRP2 heme looking at the IL-1 beta response. If you strip HRP2 again, you don't get a response. If you load the HRP2 with zinc protoporphyrin instead of heme, which is iron protoporphyrin, you don't get a response. That's actually because it's not binding to the cells. If you add ionic zinc, it will allow the HRP2 to bind but not get internalized. And I'm sorry, we're looking at surface binding here. It will bind but won't get internalized. If you use gallium protoporphyrin instead, it will bind and will get internalized because gallium has an ionic radius and structure similar to iron, more similar than zinc does. And the net result, though, is that these first couple here, the first one doesn't bind, so it doesn't stimulate interleukin-1. The second, adding ionic zinc allows it to bind, but it doesn't get in. And gallium allows it to bind and get in, but you really need the iron porphyrin for activity. And another indication that it needs to get in is that we can block endocytosis with methyl beta cyclodextran, and that inhibits endothelial activation. We can knock out caveolin-1, and we block endothelial activation. And we can block acidification of endolysosomes, and we block acidification. So we think that it needs to get internalized through caveolae and then acidified. And this acidification is very important here because if you do a heme titration of HRP2, it binds about, it's just under 20 hemes at acidic pH, and that goes up to the 50 hemes at neutral pH. So there's a pH dependence to it. And if you use HRP2, if you hold HRP2 constant and titrate the heme, or hold heme constant and titrate the HRP2, the amounts don't matter within a certain range. What matters is the ratio. And 20-to-1, you don't get activity, IL-1 beta release. 30-to-1 or above, there's a threshold, you do get activity. So what's going on here? Well, we think that there are core hemes and then there are acid labile hemes that are in play in the acetylysosome. And so what we find is that this was a clever experiment suggested by a rotating graduate student named Vibert. And what Vibert suggested is that we try mixing and matching. So 40 hemes loaded onto HRP2 causes an endothelial activation. 20 hemes does not, and that's what I just showed you. 40 galliums does not, and I showed you that. But if you first load HRP2 with 20 gallium protoporphyrins and then add 20 hemes on top of that, you get activation. And if you switch the order and load with 20 hemes first, and then add 20 gallium protoporphyrins, there's no activation. So we think that there are these basal core heme sites that need to be loaded, and then the bioactive ones are on top of that. So we think that pH is important, heme is important. The heme has to get out of the acetylysosome, and there's a heme transporter called HRG1. When we knock that out in the endothelial cells, we don't get activity. And HO1 is critical for the catabolism of heme. And usually, the HO1 is very cytoprotective for cells. But in this case, when we knock out HO1, we don't get inflammation. We don't get an IL-1 beta response. So it's the opposite of cytoprotective in this case. And so HO1 degrades the heme inside the cell. And one thing that's released is iron, which can be redox active. It can do Fenton chemistry in contrast to gallium, which is inactive. And so we can show that using a fluorescent redox active dye, as you saw earlier today, we can measure redox in the cell, and HRP2 with heme causes reactive oxygen species, just like our control TBHP oxidant. You can partially block this with N-acetylcysteine. You can block the HRP reactive oxygen species also with N-acetylcysteine. Gallium protoporphyrin does not cause this. And we're using a different assay here. We're going back to an endothelial barrier permeability assay just for detection purposes. And we tried a number of different blocking agents. Hemopexin blocks the heme, it sequesters the heme, and acetylcysteine blocks the ROS. Desferoxamine blocks the, chelates the iron, and zinc protoporphyrin is an inhibitor of heme oxygenase 1. And all of those block this, inflammatory reaction. So at the end of this, I don't have time to take you through it, but the reactive oxygen species triggers a non-canonical NLRP3 inflammasome, which generates the interleukin beta. We have evidence that that gets secreted and binds back in autocrine fashion to the IL-1 receptor, where it then can activate NF-kappa B and set off a cascade to disrupt the barrier junctions between endothelial cells, causing the vascular leakage. And one can interfere with this at different points along the way. There are caspase and inflammasome inhibitors. There are interleukin receptor antagonists like anakinra, and these all are effective in blocking the increased permeability due to HRP2. Finally, when we look at the effect of HRP2 in mice, what we find is that they cause vascular leakage. So we can infuse the mice with HRP2 and then inject a fluorescein dye and look at the brain. And what we find is that an irrelevant protein does not cause leakage. ApoHRP2 causes only minor leakage. The LPS control positive control causes leakage. And HRP2 heme causes leakage as well, while the APO-HRP2 does not. So heme is essential for the effects of HRP2 in vitro and in vivo. So what I've told you then is that HRP2, we believe, is released into the bloodstream where it binds to the endothelium, and our evidence is mostly in the brain, but there are other sites as well that we believe can be affected. It binds to the endothelium via glycosaminoglycans, gets internalized via caveolae. Upon acidification, the heme is released. That gets out. It gets metabolized by heme oxygenase. In fact, there are polymorphisms of heme oxygenase that are correlated with severe malaria. They're promoter polymorphisms that increase the expression of heme oxygenase and apparently have more severe malaria. And so HO1 is pro-oxidant in this case. You're basically releasing an overwhelming amount of iron in a short period of time. It's more than the cell can handle, and it triggers the inflammasome, which causes barrier junction dissolution and vascular leakage. That's the hallmark of severe and cerebral malaria. So this work was spearheaded by a pediatric infectious disease fellow in the lab, Sue Nguyen, with important contributions from Malaria Russo. Priya Pal started the whole project with help from two Dans and Vibert as well, and contributions from other people in the lab, from Robin Klein's lab at Washington University, helpful suggestions from Jeff Henderson, the regulatable system from Jacqueline Niles at MIT, and the help of the NIH group with the patch clamping. Thank you.
Dan, up here. Joel Vega, NIH. Beautiful work. So many questions. Let me see how many I can pack in one question.
Well, I'm happy to talk to you afterwards to ask
we will.
the ancillary ones.
So what is the affinity of HRP2 to heme compared to albumin and hemopexin? And in patients, it's been shown that hemopexin is high in uncomplicated malaria, but then it goes really low in complicated malaria. So I'm wondering if an infusion of hemopexin early in infection could help to prevent all this damage.
It is possible. We don't have a good measure of the affinity of the heme for HRP2, and it changes with the different sites as they're being loaded. So it's complicated, and we've tried to measure that, and it's tricky. But hemopexin is femtomolar, so it must be much more tight than the interactions that we're seeing with HRP2 and heme. So I think that is a real possibility.
Dan, thank you. It's really a neat story. How does this mix in with the observation of clinical isolates with deletions of the genes, and is the pathogenesis in those patients different than in others?
That's a question we would love to know the answer to. So there are strains with deletions in HRP2 and the related protein HRP3. And in fact, there's some evidence that those deletions may be an evolutionary adaptation to respond to the rapid diagnostic tests that are used for malaria. The problem is there's a disconnect between regions that have the deletions are not common in many areas of the world. Where they are common, the question is there enough severe malaria? We need to find a population with enough severe malaria and enough HRP2 deletions to see whether there's a correlation, anti-correlation. We are talking with people in Ethiopia where there may be enough of both to actually ask that question. The anecdotal reports are that the deletion patients don't have very severe malaria, but that's, I wouldn't put much stock in the anecdote so far.
Dan, great talk. Question about how actually the HRP2 gets loaded with heme and where does it happen? Because this is actually secreted into the red cell, they're not likely to have that high concentration of free heme over there. And second, is it oxidized or reduced heme? Fe2 plus or Fe3 plus heme that you're seeing over there?
Well, I'll answer the second question first. We haven't assessed the state of the heme when it comes out of the parasite on HRP2. We load it with hematin when we reconstitute, and that's Fe+3. But of course, there are redox mechanisms that are active. In terms of where it gets the heme, there's actually, according to the literature, there's quite a bit of free heme in the red blood cells, micromolar amounts of free heme in red blood cells. And it's coming on and off of hemoglobin in equilibrium, and it's in excess. And there's heme there. There's also heme in the bloodstream as malaria progresses. It's possible, though, that in patients, we haven't had enough blood to fully assess this, that in patients there are different levels of loading of heme that could make a clinical difference. And we'd love to know about that.
Thank you so much for such a great talk. I really enjoyed it. My question is related to the second question, that what is PFHLP actually doing for the parasite? I really enjoyed the how it's causing the clinical phenotype, but although it's so abundant, I'm not sure what it's exactly doing for the parasite.
Right. Well, we don't know. Some of it is intracellular, but it is not essential, and parasites seem to grow well in culture without HRP2. But one of the things I didn't show you data for, but the HRP2, as it's interacting with the endothelial cell and causing inflammation, it's causing upregulation of surface receptors on the endothelial cells, ICAM1, CD36, et cetera. And those are, as Kirk showed earlier, that's so that the parasites can adhere better and avoid the spleen. So our speculation would be that it's not doing it to cause devastating vascular leakage, it's doing it to cause inflammation of the endothelial cells for cytoadherence.
Thank you so much. Sorry, I didn't introduce myself. My name is Tomaki. I work here at the Hopkins.
Yeah, one quick question, Dan.
Where are you?
I'm way over here.
Okay.
You started your, this is a follow-up, I think, of other questions. You started your discussion showing a very dramatic difference in the amount of HRP2 in uncomplicated versus severe malaria. What I wanted to make sure I understood, do you know that qualitatively they are the same in terms of heme conjugation to the HRP2? And is their turnover rate, is their half-life in serum similar?
Interesting questions. We do not know whether their heme loading is the same. And I think that would be very interesting and important to know. And I'm sorry, the second part of your question was?
Just whether the turnover was
The
similar,
turnover.
whether they were equally stable.
Yeah, we don't have a good assay for that. There, of course, is the assay for bulk HRP2, and it is very slowly turned over. You can detect HRP2 for up to a month in the blood of patients with malaria, so it certainly does linger, but whether the heme loading changes or not, we don't know. It also lingers in the endothelial cells, so after several days, we can still detect it. HRP2 doesn't have many protease cleavage sites in its sequence, and it probably just hangs around in the cells that take it in.
Thanks.
Thank you.